continue our discussion of the paramecium Aurelia species by diving into its morphology. So as we all know, paramecium Aurelia are going to be eukaryotic, unicellular, I'm very sorry about that. They're eukaryotic and unicellular protists that are but micrometers in length. Now, they tend to be about 150 micrometers long and 50 micrometers broad, but that can vary, and that can depend on the food supply, the stage of development, the strain, and the conditions they are in. Now, for example, we have the paramecium Aurelia complex, which typically thrive in extreme environments. And as a result, they're going to be a little smaller and they're going to have more specialized functions that we're not really going to dive into at the moment. I'm going to dive into getting the fundamentals down. So we have what's known as the pellicle, which is going to be the coating of paramecium. Now, it's going to be a thin gelatinous substance. It's going to be produced by the cell, obviously, produced in the cell and secreted outside to form this membrane. It's and define its shape and form a sort of protection. Now, the paramecium Aurelia complex has an elaborate structure, as you can see in this photo above. I can see a photo outlined here, and you can see the alveolar membrane, the alveolar outer membrane, the inner membrane, the cavity. And outside of that, you're going to have the cilia forming. Now, the cilia are going to consist of these fibrils that are, they're going to have one fibril on the axis and anywhere from 9 to 10 of those fibrils dispersing, thousands and thousands of cilia. And you have two kinds of cilia that you have typically the oral cilia, which is used to feed, and you have the body cilia, which is used for like locomotion and conjugation to reproduce with other cells. And you have what's also known as trichocytes, which is going to be these as you can see, they're parallel to the cilia, and you can see the opening. And now these are going to be discharged as filaments, and they're typically used to repel against any predators and to protect the protist from any other external force. And you also have the kinetostigma, which is going to be where, to create more of a fortified structure for the paramecium complex. And inside the cilia, as you can see, this is the morphology of the cilia. You can see how it forms on the very outside of that protist and creates a sort of bean shape curve. And you can see microscopes and how scientists have further discovered what's inside cilia, which is going to be these microtubule bundles, and there's going to be protein fibers inside of that. And these protein fibers are going to create a highway for the transportation of molecules and organelles, I'm sorry about that, inside and outside the cells. And this is also going to be very important in reproduction, which we're going to talk about in later discussions. And these motor proteins are going to be called dynein, and they sit across each paired microtubule, and they use ATP as energy to really propel this locomotive force that are inside cilia. Now, it's going to be ciliary movement is typically determined by water and attracting water. You're going to have an effective forward stroke and a recovery stroke to preserve it, which is going to be very similar to how we swim. But paramecium can have thousands of cilia at a time, so they're not going to be like as simple as just us moving back and forth. So cilia are going to they're going to have like this rhythm, this biological rhythm where cilia that are transverse or like on parallel opposite sides of each other are going to move at the same time. And then you can also have what's called a metachronous rhythm, which is where the cilia of like a longitude or like a row beat one after another. And it's going to create waves passing, propelling locomotion as well as the movement of water from the posterior end to the anterior end.
So the paramecium Aurelia has some really amazing organelle components that are quite unique for paramecium itself. It contains many of the same cellular components as eukaryotes, such as mitochondria, but also has some really unique organelle structures. So the very first layer of paramecium is called the pellicle. There's a layer under that is composed of a somewhat viscous type of cytoplasm called ectoplasm. This region contains spindle-like organelles called trichocysts that aid in the discharge of waste from the cell, as well as movement and shielding and repelling the cell from attacks from outside sources. Below the ectoplasm is called the endoplasm, which is a less viscous layer that contains the vast majority of the cell's organelles, including vacuoles, lysosomes, mitochondria, the nuclei, rough ER, smooth ER, as well as ribosomes. The nucleus is the most unusual part of the paramecium Aurelia, or I should probably say nuclei, as they have three, which are all different types of shapes, contents, and functions. The first two are called micronuclei, and the other is called the macronucleus. The micronuclei contain diploid copies of DNA, whereas the macronucleus is polyploid, containing multiple subsets of DNA as well as almost thousands of copies of the DNA itself, which makes the paramecium Aurelia's trinuclei complex so unique compared to other paramecium within its family.
The epiplasm is a superficial cytoskeleton layer of variable thickness that both surrounds basal bodies and interacts tightly with adjacent membrane networks. The paramecium Aurelia does not have a cell wall, since it is a single-celled protist. Instead, its body is covered in cilia, which are hair-like structures. Also, paramecium Aurelia has pellicles, a colorless cell membrane. And in the diagram that I have given here, you can see the pellicule, which is around the outside of the cell, and then the cilium coming out of it, which are the hair-like structures.
Cells swim and feed by beating their thousands of cilia in coordinated patterns. The organization of these patterns and its relationship with cell motility has been the subject of a large body of work, particularly as a model for ciliary beating in human organs where similar organization is seen. However, the rapid motion of the cells make quantitative measurements very challenging. So two distinct values of CBF, or ciliary beating frequency, appear at different regions of the cell. Most of the cilia beat is in the range of 15 to 45 hertz, or Hz, while the cilia in the peristomal region beat at almost double the frequency. Moreover, the measurements do not display a measurable correlation between swimming velocity and the mitochondrial wave velocity on the cell periphery.